Okay. All right, I see answering the questions in the book. I would agree sometimes the questions in the book can be rather tricky. Counting all the seeds, that definitely is a very tedious part of the lab. I don't know that it was super difficult, but it definitely was a tedious part of the lab. So, okay, thank you very much for that feedback, guys. Now, up on your top right hand or your PowerPoint screen, I went ahead and I put your actual lab reports. I took bits and pieces off of everyone's report and kind of merged it into one. So the first thing is you all had a great title, a very simple title. We don't need anything fancy. But something that every single one of you failed to do was put your name on the lab report. So make sure you put your name up there, and I may not have added it to my checklist, so I apologize for that, but your name needs to go on your lab reports so that I know whose I'm grading. Now I know when you submit it, I can tell where it is, but make sure your name goes on your lab report. Now, then the next thing is I had the different sections: materials, procedure, results, conclusion. I really was looking for either a bolded, an underlined, a highlighted word that says materials with a list of the materials you used. Okay, and honestly, I don't remember whose lab report I took what from, but whoever this was a very good materials. Nice, simple, white paper, pen, seeds, lab manual. That was everything we used. Another way you could have written this was as a bulleted list, columns. That would have been fine. Or if you wanted to, you can go ahead and put the heading materials and then write, in this lab, we used, and list what you used: white paper, pen, seeds, and a lab manual, period. Either way is fine. My personal opinion is the simpler, the better. So I would just list it right like this. The next step was the procedure. Again, bolded, underlined, whatever, the word procedure. And then this person chose to write steps. If you are writing steps to the procedure, you don't have to write the word step and then the number 1. You could just write the number 1. But if you want to write the word step, that's fine. If you are using steps or numbers, each number should be its own start of a line. So like here, step 1, she wrote step 1, or he pressed enter, step 2, pressed enter, step 3, so that it's an easy list to read. This was a great procedure. On the white piece of notebook paper, I drew with a pencil a diagram with one main bubble at the top, branching out to 2 other bubbles to the right, bubble A, and left, bubble B. I then drew 2 other bubbles branching out from each of these 2 bubbles, bubble C and D to bubble A, bubble E, and F to bubble B. Branching out from bubble C is bubble G. Branching out from bubble D, there are 2 bubbles, H and I. Branching out from bubble E is bubble J. Branching out to the bubble F are bubbles K, L, and M. Certainly a tongue twister, but it's a very good description of what you drew on your white sheet of paper. Another way to have written this is to insert a picture. In your lab book, it had a picture and it said like figure 2-1. You could have written step 1 on a white piece of notebook paper, copy figure A. And then you would insert a picture of figure A into your procedure. But this was done correctly. Now, one thing that should not be used in a lab report is the pronoun I. So instead of saying on the white piece of notebook paper, I drew, you should write on the on the white piece of notebook paper, draw. because you're not telling people what you did. You are trying to tell people how they can do the lab. So think of it like this. You should be able to take your procedure, hand it to your brother or your sister, and they should be able to do exactly what you did, just by reading your paper or looking at pictures. So we don't want to use the pronoun I. Okay, but this lab report, this procedure was very nicely written, very, very detailed. It was a great job. Okay, then in the results, you guys answered the questions, which were a bit tricky. The comment that I have is when you're answering a question, you need to answer it in a complete sentence. Just writing yes, it looked nice, doesn't work. Okay, you guys are in high school, you need to write in complete sentences. Keeping them numbered is fine. You could put number 1, the answer to number 1. Number 2, the answer to number 2. There is absolutely no reason for you to write out the questions. I have a lab book sitting right in front of me. I can read the questions. Okay, so just answer the questions in complete sentences. And now there were 2 different approaches to sending me your data. The first was a hand-drawn chart. And it's hard to read the numbers because I made them smaller so they would fit on the page. But you get the idea. It was a hand-drawn chart, just like we did in class, and a picture of it. Perfectly fine. Someone else decided that they were going to do a computer version using, I don't know if it was done in Excel or Word, whatever. But that is fine as well. So both of those were great ways to send me your results. All of the lab reports were very, very nice. And Anthony, that's perfect. That's why I'm showing you guys this. I'm showing you exactly for the comment that Anthony just made in the text chat. Why didn't I think of that? A lot of times when we're doing science or we're doing anything, we're thinking in one frame of mind. And whoever drew the drawing might be thinking, oh, the only way I'm going to be able to do this is to draw this. And the person who did it on the computer might be thinking, oh, the only way I'm going to be able to do this is to draw on the computer. When in reality, there are lots of different ways to do the same or present the same data. I have also had students set up using, in Microsoft Word, there's the flow chart. I don't know if it's an icon or the flowchart template. That's the word I'm looking for. The flowchart template, where it will pre-do the circles for you and the lines connecting it. That would have been perfectly fine as well. There really is no wrong way to present the information. I've even had students present it in a table. Now, a table does not look anywhere near as neat as the diagrams you guys have here, but will still work. So you should have gotten your lab reports back via email today. I sent it to the emails that were on your account in Caselli. So if for some reason it didn't go to you, check with your parents to see if they got it in their email. And if for some reason they didn't, give me a call, send me an email, and I'll send it back out. The rewrites are going to be due to me the same time your next week homework is, which is next Wednesday. So under week 1, there's going to be a spot that says lab resubmission, and that is where I would like you to put, the resubmitted lab report, where I will grade it. I will make further comments on it if necessary, but I truly think that you guys will all do fantastic on it because you did do fantastic the first time around. Anyone have any questions that I didn't go over about your lab report, either on a specific comment that I made on your lab report or just in general? When I made the comments, I made them right in the document by using the comment tab. So when you get your Word document from me, you're going to have to, from across the top in Microsoft Word, choose Review. And that's where you should be able to see my comments if they don't show up right away. And if for some reason you're having a problem getting them to show up, give me a call, send me an email, and I can make a PDF of my comments for you. So anyone have any questions on the labs or the lab report? No? All right, fantastic. Well, we have two things that we are going to cover today. The first thing is how to make scientific drawings, and the second thing is cells. So it is completely up to you guys which we do first. I need to cover both today, but the order does not matter to me. I will tell you that the scientific drawing part is a little bit more interactive than the cell part. So if you guys could, on the tab I just sent you, let me know which you would like to do first. Your choices are cells or scientific drawing. We are doing both today, but whichever one gets 2 votes, we'll do first. And by unanimous decision, we are going to do scientific drawing first. Definitely a fun choice. For this, you are going to need a pencil, preferably pencil, paper, and probably an eraser. And maybe a ruler or a straight edge, but it's not that big of a deal if you don't have a ruler. So if you look up on your PowerPoint screen right now, we're going to look at a PowerPoint that says scientific drawings. Last week we talked about making observations, and we made some verbal observations. Well, a lot of times in science, and biology in particular, you need a visual observation. There's lots of reasons for this. A visual observation allows you to describe something to either your reader or to someone else in more detail. For example, I could say, my cat has 4 legs and a bushy tail and some whiskers. And that's great, and you probably can picture it because it's a cat. But how big is the cat? You know, and last week we used the ruler to help us with that. Or how bushy is bushy? And this is even a bigger advantage if you can do scientific drawings when you're looking at microscopic things. So we are going to make some microscopic drawings. When you are making drawings, you need to look carefully. Accuracy counts. For example, and I'm just going to draw a rough sketch of the face of my cat. Now, keep in mind, I am not, in my opinion, a good drawer by any means. So go ahead and look at your screen, the video screen, and you can see maybe. There you go. You can see the face of a cat. Now, a more accurate picture would actually be if I drew her whiskers more to scale. My cat has very, very long whiskers. Now, there are 2 pictures that I'm going to put up on the screen, picture A and picture B. Picture B is a much more accurate picture. It gives you a better idea of how long the whiskers on my cat are. So accuracy definitely counts. When you are drawing a scientific drawing, you want to get the big picture. You start by drawing large structures, and then you add the details in later. If you have a picture with lots of details, you are going to want to make your drawing as large as possible so you can add the details within reason. If you are writing a lab report, you are not going to put the picture of the onion cell on a huge piece of poster board. Okay, it needs to fit in your lab report, so it would just go on a normal 8.5 by 11 sheet of paper. You need to label your drawings. Okay, so if we look back at my cats, these are not labeled. So we need to add labels. Using a ruler is the neatest way to do that. So I just went ahead and added 2 labels to diagram B. I added ears and eye. We need to add a title. Your title should be as specific as possible. So, and as neat as possible. So I am labeling this Mrs. Berg's cat. You need to add a perspective or a description of how big the object is. So this is where a ruler comes in. If you are drawing something that you can measure with a ruler, you measure it. So for example, the width of my cat face is 7.5 inches. So in order to label that on my diagram, I drew brackets from the right side of the face to the left side of the face, and underneath the brackets, I put the width. You do not need to measure every part of your picture. I'm not going to sit here and measure my cat's eyes. First of all, she wouldn't let me. Second of all, you know it's going to be relative to the size of the picture. If instead of 7.5 inches, I put the width of my face, my cat's face was 18.5 inches, now you may be thinking Mrs. Berg has a lion at her house, not a cat, because the size is so much bigger. So scientific drawings are meant to show you details in the picture, and they are meant to give you perspective. So up on your screen right now, this is a picture of a volcano. What is missing from this scientific drawing? Who would like to share? Raise your hand. What is missing from the scientific drawing up on your screen? I'll give you a hint. With my laser pointer, one of the things should go up here. Alicia, go ahead.
A title.
Awesome. The title is missing, and there's something else missing. On my picture, it was down here at the bottom. It's missing the title and something else. Anthony, go ahead. I saw you hit the button, Anthony, but I wasn't able to hear you talk. Let's make sure your microphone is on. Go ahead and look at the bottom left-hand side of your screen. You see a wrench. Click the wrench, and a pop-up box will open up. Choose microphone settings. Make sure the correct microphone is selected. Make sure that your microphone's not muted. For example, on mine, I have a button on the cord that I can mute with. So go ahead and Anthony, let's try that again. What is missing? In my picture, it was in the bottom.
The name.
Okay, your name would be a good thing to have on here as well. Very good. And Alicia put in the text chat perspective. The size. Is this volcano that they didn't label with a title, is this volcano 2 inches wide or is it 2,000 feet? And that's important when we're doing scientific drawings. What I would like you guys to do is just on a scrap piece of white paper, I would like you to make a scientific drawing of something in the room you're sitting in. Now, you may not have anything biological in the room you're sitting in. Make a scientific drawing of anything that's in there. It could be, I'm sitting in here and I see my bookcase. It could be a bookcase, add a couple labels. I have an easel in here, it could be that. Make a scientific drawing that we're going to turn our cameras on and share with each other of something in your room. Right now, my computer says it's 12:24. When you are done, I want you to click done on the poll, but you have until 12:30, so you'll have 6 minutes if you need it. Okay, you have 2 more minutes. I see that Sarah is done, but there's 2 more minutes to finish your drawing. Okay, 1 more minute. All right, I'm going to go ahead and stop you guys there. If you are done, you can show us your completed picture. If you're not done, you can show us what you have. I will show you my picture first. I will caution, it is pretty hard to get things centered on your video screen. So when it's your turn, just make sure you kind of look what you're doing. So here I have my picture. I have my title as fish. My name is on the bottom. I have my labels of dorsal fin, caudal fin, anal fin, and gill. Now, depending what I was looking for, I may have different things labeled. For example, I may have the eye of my fish labeled or scales on my fish labeled. So you're not necessarily going to label every single thing in your picture. On the bottom, I have 2 squiggly equal signs, which means about 3 inches, because I did not take a ruler and put it in my fish tank. And on the right-hand side, it's about 1.5 inches tall. So there is my diagram. Who would like to volunteer to show us their diagram first? Any volunteers? Awesome. Okay, Alicia, so I just turned your camera on. Now, underneath where my video normally shows up, you should see a button that says Start and Stop. You need to click Start to turn on your camera. And here it comes. I'm going to turn my mic off so you can turn yours on. We can see you. Perfect.
Hello! Okay, I don't really have anything biological in my trailer, so I drew a coffee cup, and you can kind of see how at the bottom its width is about 4 inches, its height is about 9 inches, and I labeled the handle and the lid. And there's a glass container, there's a plastic green holder, and there's a metal rod inside of it, and then there's the spout. Yay!
Awesome. Thank you so much for sharing your picture with us. It's not a big deal to not have something biological. It doesn't matter. It's still a scientific drawing, whether it's a biological scientific drawing or not. So great job with all of your labels, with your bib or your size, and you definitely are a better artist than me. All right, Anthony or Sarah, who would like to go next? Any volunteers? Remember, part of your class grade is participation. Oh, I have to turn it on your end. Okay, Sarah, so what you need to do is underneath where my video normally shows up, you should see a button that says start and a button that says stop. If you go ahead and click start, your camera should come through. Oh, I see you. Perfect. I'm going to turn my mic off so you can turn yours on.
I have a computer, and it's just white, and I measured how tall it is, and how wide it is, and what colors, and where the bottom is, where the top is, the title. Yeah. That's that.
All right, fantastic. Thank you very much, Sarah. Great job. Anthony, can I go ahead and turn your camera on now? All right. Your camera's on. Underneath my video screen, there should be a button that says start. Go ahead and click the start. Perfect. I'm going to turn my microphone off so you can turn yours on.
All right, so I just have a simple picture of a chair and the dimensions of the seat and the back and the legs. That's pretty much all I have.
Awesome. Great job. Thank you very much, Anthony. Thank you all for going on and sharing your pictures. You guys are all very good artists, and I'm very impressed. And here up on your PowerPoint screen, I went ahead and here is a big drawing of a ladybug, something that's much smaller than the picture you drew. So Anthony in his picture drew a chair. Obviously, he had to draw this chair smaller than a live chair. If he drew the chair to scale, he would have needed a much larger piece of paper. A lot of times, scientific drawings are done like that to make larger things smaller, or to go the opposite way and make smaller things larger. So this ladybug is not drawn to true size. It is drawn larger than life. However, they are missing some things. What is something that they did not include in their scientific drawing? Go ahead and raise your hand or type it in the text chat. What did they not include in their scientific drawing? Very good. There is no title. There is no perspective. There are no labels. Okay, so they really didn't write anything on here. They only did the illustration. So, fantastic job on the scientific drawing. We are going to switch gears now, and we are going to talk about cells. Your homework this week is going to be a combination of scientific drawing and diagrams and cells. So we are going to take a look at cells. I know most of you have looked at cells in your actual biology class, but we want to take a look at a few specific parts of cells. We are going to start by looking at an image or a diagram of cells. So up on your PowerPoint screen, I have an animal cell and a plant cell. Now this is not necessarily a good diagram. The writing, unfortunately, is pretty small to read. But if you go into Moodle, there is a much better, it's actually the same diagram. It's just much larger. So you can compare the differences between plant cells and animal cells. Now what I would like to do to get started is we are going to make a Venn diagram on our whiteboard. So we are going to have a plant cell and an animal cell. And then the part that is highlighted in yellow is where the characteristics of both plant and animal cells will go. So if it's a characteristic of a plant cell, you put it in this part of the circle. If it's a characteristic of an animal cell, you put it in this part of the circle. And if it's a characteristic of both cells, you put it in the middle yellow area. So go ahead and take a minute and fill out, by writing on the whiteboard, some characteristics of plant and animal cells. Okay, make sure you're looking at the right side. And the plant characteristics in the plant side, and the animal characteristics in the animal side. Vacuole or animal cells do not have chloroplast. No problem, that's what I figured, Sarah. Not a big deal whatsoever. So I went ahead and I added a couple of things up here. I added on the plant side that they're usually square or rectangular shapes, and on the animal side that the shape varies. Plants also have a cell wall, a vacuole, and chloroplast. for characteristics that are common between the two, we have DNA, cell membrane, nucleus, rough endoplasmic reticulum, ribosomes, Golgi apparatus, smooth endoplasmic reticulum, mitochondria, lysosomes, cytoplasm, nucleolus we could have up here. We could have cell membrane up there. And then, an obvious thing that we could have put up here is plants. Cells are found in plants. Animal cells are found in animals. Okay, you guys did a great job on that. And now we're going to take a look at some information in a PowerPoint about cell structure and functions. So cells are the smallest living unit. Most cells are microscopic. Here is an example of an animal cell on the left and a plant cell on the right. Now, some of the lab kits, or the lab kits that you guys have, all of you, have microscope slides in them. If you have a microscope at home, you are certainly more than welcome to look at the slides. However, it is not a requirement of this class to buy a microscope, so do not feel like you need to go out and buy one. I am going to show you lots of microscopic slides today and over the course of the class, right up on your PowerPoint screen, so you don't have to purchase a microscope. Cells were discovered by Robert Hooke in the mid-1600s. He was observing a sliver of cork, like that would come out of a wine bottle or that may be on a cork board in your bedroom. And he saw what he called rows of empty boxes. Now, he coined the term cell for these empty boxes, and that is how cells were discovered. The picture up here that is dyed in pink is a sliver from a piece of cork underneath a microscope. Now, something that's missing from this scientific diagram is a size. If you're looking at something microscopic, you should label it 10x, 100x, so we know how many times the magnification is. The cell theory is something that you probably have heard of. In 1839, the cell theory was first thought about by Theodor Schwann and Matthias Schleiden, and their version of the cell theory is that all living things are made up of cells. Simple enough. 50 years later, Rudolf Virchow added to that cell theory and said that all cells come from cells. That was his addition. The major principles of the cell theory is that all living things are made of cells. The dissections we do at the end of the class, all of those organisms are made up of cells. The trees outside are all made up of cells. A cell is the smallest living unit of structure and function of all organisms. All cells arise from pre-existing cells. This idea discounted the idea of spontaneous generation, that a cell could just, poof, show up. All cells have to come from other cells. And there's a large range in cell size. This diagram up here is meant to give you an idea of different cell sizes. Okay, so in the top left, it starts with a typical plant cell. Okay, in a typical plant cell, the size range is from 10 millimeters to 100 micrometers. And then you have a hen's egg, which is a single cell. It is 65 millimeters. You have neurons. You have human red blood cells up here. You have E. coli, which is a bacteria up here. You have viruses, which aren't cells, but I put them up here for size-wise. Cells have a very large area to volume ratio, and it's because of their shape. This diagram looks at the area to volume ratio just a little bit. And the area to volume ratio, if you have a cell that is 2 centimeters by 2 centimeters by 2 centimeters, the area to volume ratio is 3 to 1. Now, if you have 8 cells that are 1 centimeter by 1 centimeter by 1 centimeter, and the ratio is 6 to 1. Now, that's important when you start talking about how cells move materials through their body, which we will do in a couple of minutes. But first, I need to look at the characteristics of cells. Okay, cells have a surrounding membrane. Inside of this membrane is a protoplasm, which is a cell that has a very thick fluid. There are various organelles, lots of which you guys mentioned when we were comparing plants and animal cells. And there's a control center with DNA. Now, what we were going to do right now is look at prokaryotic versus eukaryotic cells. Prokaryotic cells were the first types of cells on Earth. They are the cell types of bacteria and archaea. Prokaryotic cells have no membrane-bound nucleus. Instead, they have a nucleoid. A nucleoid is a region of DNA concentration. Organelles are not bound by membranes. And eukaryotic cells have a nucleus bound by a membrane. They include fungus, protists, plants, and animal cells, and they possess many organisms. Here's a representative animal cell. I'm not going to read all of these to you. I just want you to take a minute and look at some of the organelles in an animal cell. And here's a representative plant cell. Let's take a look at this and try to think in your mind a comparison between the two. Okay, and we're going to just really quickly fly, like super fast through all of these organelles. If you have a specific question on one of them, certainly go ahead and stop me. But that's something that's easy enough for you guys to look at in your biology book. But I do just briefly want to touch on all of these, most of these. So there are some bacteria-like organelles. Oh, hold on. Wrong one. Okay, this is what I wanted to do. Okay, so we're going to talk about the plasma membrane, which is the outer area of the cell. Inside of the plasma membrane is all of the cells, all of the organelles, all of the cell contents. This plasma membrane is a double layer of phospholipids and proteins. Now, this can be a concept that's sometimes a little bit difficult to think about. Okay, if you look at the diagram up here in the top right, you have on the top a watery environment outside the cell. On the bottom, you have the watery interior environment of the cell. And in between, you have the plasma membrane, which is made up of phospholipids, which are fats, carbohydrates, cholesterol, and proteins. Now, in order for the cell to work and function properly, some objects need to pass through this membrane. But not everything can, because if all of a sudden the nucleus ends up on the outside of the cell, you have a problem. So here is a close-up of a phospholipid, which is what the cell membrane is made up with. You have a hydrophilic head and a hydrophobic tail. Hydrophilic means water-loving, and hydrophobic means water not loving. So the hydrophilic head points to the outside of the cell, and it interacts with the water. If you look at the left of this diagram, you see one phospholipid that is labeled. Glycerol is the blue head. The kink, or the tail going down, one of them has a kink or a turn in it. And the two tails are made up of fatty acids. So some molecules need to move freely across a plasma membrane. Some molecules that move freely are water, carbon dioxide, ammonia, and oxygen. And some molecules need help to get across the plasma membrane. The molecules that need help use carrier proteins. And the carrier proteins transport the molecules through the plasma membrane. Most of the time, these proteins are embedded in the lipid bilayer, and they form almost like channels through the plasma membrane. So if you think of this on a much larger scale, and you think of a cell as a harbor that boats would go into. Some things can move freely in and out of the harbor. Carbon dioxide, oxygen, there's no barrier that stops that. But there could be either land or rocks like a jetty that keep other molecules from moving freely. Boats can't just move in and out of the harbor. They need to find a channel or a carrier protein so that they can move over or through the membrane. Okay, and here are a few examples. You have your plasma membrane. You have A, small uncharged molecules that move freely. In B, you have lipid-soluble substances, that means fat-soluble, that move across. You have water-soluble substances that do not move across. And then you have ions that do not move across.